So today we have Elle Braverman presenting on atherosclerosis. She's a first-time presenter, so clarification questions only. And here she is.
Hi everyone. Today I will be presenting on atherosclerosis. So, what is it? Atherosclerosis is a disease where the arteries become clogged or hardened by plaque. That is why atherosclerosis is more commonly known as plaque buildup. This disease can affect any artery in the body, but it is most notorious for its effect on the heart. Atherosclerosis leads to cardiovascular disease and is the cause of most heart attacks. Unfortunately, heart attacks are incredibly common in our society. Every 43 seconds, someone in the United States suffers from a heart attack. And this is so common because there are only two arteries, the left and right coronary arteries, that provide oxygen-rich blood to our heart. And a heart attack occurs when even just one of these arteries are slightly blocked or clogged down by plaque. And this blockage by plaque is atherosclerosis. Atherosclerosis is found in 80 to 90% of Americans over the age of 30. And current treatments really only include changes in diet and health and medication to alter cholesterol and blood pressure. However, these therapies only reduce cardiovascular events by 20 to 30%. So my paper investigates a possibly more effective way to stop atherosclerosis. My paper is titled Non-Lethal Inhibition of Gut Microbial Trimethylline Production for the Treatment of Atherosclerosis. Or, in simpler terms, safely stopping plaque buildup in the arteries as a treatment of atherosclerosis. Many of you probably know that meats and other foods high in fat have been found to be bad for our health and may potentially lead to the buildup of plaque. Choline is a structural component that is found in these fatty foods that specifically lead to plaque buildup. However, meat and choline is not directly linked to atherosclerosis. There is really a so-called hidden pathway that connects these two. And the paper that I will be presenting on investigates a way to stop this pathway. Recent studies have found TMAO to be associated with plaque buildup and a part of the atherosclerosis pathway. Researchers then backtracked and asked themselves what causes TMAO, and they found that TMA causes TMAO. So really, meat and choline lead to the production of TMA, which leads to the production of TMAO, which then eventually leads to plaque buildup. So, the researchers' first step was to identify an inhibitor to stop TMA formation in order to stop the entire pathway. As I said earlier, choline is believed to cause TMA, so the researchers used choline and choline TMA lyase activity to find an inhibitor. Researchers tested different choline analogs on E. coli cells, and they measured TMA production and found two choline analogs that showed an inhibitory effect. And these two were DMB, which is shown in red, and TMSI, ETOH, which is shown in blue. So these two were able to stop TMA production. And the study continued with DMB because it is found naturally in certain foods. So the researchers hypothesized that this would be less toxic to our body and cells. The researchers then wanted to further verify that DMB did in fact inhibit TMA. So cells were grown with or without DMB. The researchers then measured TMA activity, and the cells that were grown with DMB had much lower rates of TMA. So this was just verification that DMB inhibited TMA from live microbes. The second step the researchers took was to determine if DMB is lethal. So if DMB is to be used for a possible therapy, it can't be lethal to our cells. So cells were grown in a nutrient broth with or without DMB. The researchers then measured the growth rates of both. And as you can see here, the growth rates in both of them were exactly the same. So the only difference was that DMB, as expected, had lower levels of TMA. So yes.
What kinds of cells were they growing?
They were P. mirabilis. Or E. coli cells with P. mirabilis, which just contains an enzyme that catalyzes TMA. So TMA is the first step of the atherosclerosis process that DMB was able to inhibit. The researchers then wanted to see if DMB can stop the second step, which is TMAO. So mice were placed on either choline or carnite diets, and these diets are previously shown to raise TMAO levels and accelerate atherosclerosis. And half of these dietary groups was given DMB through their drinking water. And as expected, researchers found that the choline and carnite diets, shown in blue, raised TMAO levels in mice. But with DMB, the TMAO levels were significantly reduced, and this is shown in red. So, DMB was able to successfully inhibit TMA, TMAO, and the next step the researchers took was determine if DMB can stop plaque. Foam cell formation is just an indication of plaque buildup. So, the researchers studied the effect of DMB on foam cells. Atherosclerosis-prone polyprotein mice were placed on a regular chow diet or a diet with the addition of choline, and DMB was added to both of these diets. And as expected, choline raised the amount of foam cells in mice, which is shown just in the red. But with the addition of DMB, the percentage of foam cells found in mice was significantly reduced, and this is also shown in red right here. So, the final step the researchers took was to determine if DMB was able to actually decrease plaque. So, mice were placed on the same previous diets, the diets with choline or just the regular chow diet, and DMB was again added to both. And the addition of DMB was able to significantly reduce, just shown here, was able to significantly reduce plaque and artery damage. After mice were on the diets for 16 weeks, the researchers sliced and stained aortic root sections of the mice, and mice on the choline diet had this increased buildup of plaque, which is just shown as the highly concentrated red area. But with DMB, as you can see, there is no highly concentrated area of red. Can anyone tell me what this means? Yes, Jen?
It means that there's no plaque buildup when you have DMB.
Yes. Great job. Right. So, in conclusion, DMB was able to successfully stop the process of atherosclerosis. It was identified as an inhibitor of TMA formation. It was able to lower TMAO levels. DMB reduced foam cell formation, and DMB finally was able to stop plaque buildup in aortic arteries. And also importantly, the DMB was shown to be a non-lethal inhibitor to our cells. Well, not really our cells, mice cells. So, every once in a while, we hear events about the guy who's in great shape, eats incredibly well, works out all the time, but still suffers from a heart attack. And this study provides a possible reason of why this occurs. Food is not what directly affects our health and the buildup of plaque. Really, specific pathways serve as intermediaries between food and the health of our bodies. So this study provides a possible treatment for atherosclerosis and possibly other heart diseases by stopping these specific pathways. Thank you for listening. Any questions? Yes.
So it looks like in this study they were giving
DMB.
TMB to mice before they had any sort of plaque buildup in their arteries, right? So they were young mice when they started the study. Is that
Right.
true?
Yes.
Did the researchers talk at all about how it might, because I imagine if we were applying this to people, you would already have some plaque buildup going on before you might begin treatment like this. So did they mention anything about how could this potentially help people who already have some plaque buildup?
So I mean, I think just the fact that the mice were on the choline diet for 16 weeks. So I think that kind of like simulates like the amount of plaque buildup a person would normally have. I mean, not exactly like the correct alignment between people and mice, but like that kind of accounts for the normal foods and plaques that we would have already been eating, and then DMB was also added to this.
Right.
So there's no they don't talk about specifically how this therapy would work in humans or anything, but I think it also, with the already built up of plaque, it would be able to decrease it.
Wait, can you go back to the slide where it showed the where it measured the aortic lesions? That one.
Yeah.
The one with the yeah, that one. So this is all in is this the same mice? The same mouse?
No, this is so there was 4 different dietary groups, and this, I think it's a mouse from each of the different dietary groups.
So in the 2 that don't have the concentration of the plaque, but there's no red concentration, so they don't have the plaque.
Yeah.
They were never on the diet with just the shower, just the choline.
No.
Choline.
They were on the diet with choline plus DMB.
Okay.
Yes.
When they were doing the screen of the different drugs, where we did the line crash,
Yeah.
Did they mention why this was a drug of interest, DMB, or the other one? Or kind of how they came to these, or did they do a screen of many different compounds?
So they studied like the cleavage of certain like CNN cleavage, and DMB also had the same the same, I guess, system of working. I wasn't really too sure on what they were talking about, but they just kind of assumed that probably yeah. Yes.
You might not have come across this, but did you see anywhere treatment by changing gut microbe?
No. Wait, I'm sorry, what? I don't understand your question. I'm
Okay,
sorry.
so it's the pathway is from food to gut microbes
Yeah.
to plaque, right?
Yeah.
And so I was just wondering if there have been any studies in which they switched to maybe a more vegetarian flora gut microbes to see if.
I do not know. They did not mention that in my paper, but that's something that they should look into. Yes.
So, this study, very interesting. Did they talk about how they were looking to apply it to the next step, meaning taking this particular drug or drugs like it and apply it to higher models closely related to humans?
So they did not specifically talk about how this drug could be used in humans, which is a next step that they do need to take and see how altering these different levels would affect other aspects of our health. But the paper did in fact talk about how stopping the production of TMA could alter our gut microbiome, which is something that would not be good. And also, our bodies could develop possible tolerance to this drug if it was administered.
fluctuates in order